Most people think of cloves as a background spice, something added in tiny amounts for aroma, something optional, even decorative. Yet when scientists analyze cloves at the molecular level, a contradiction appears almost immediately. The biological activity concentrated inside this small dried flower bud is disproportionate to its size, its culinary role, and the attention it receives in modern nutrition. In laboratory measurements, cloves consistently behave less like a flavoring and more like a dense biochemical signal. They interact with oxidative systems, inflammatory pathways, microbial balance, and metabolic regulation in ways that challenge the assumption that spices are nutritionally insignificant. What makes this especially interesting is that these effects do not come from novelty compounds or exotic processing, but from mechanisms that have been quietly studied for decades. To understand cloves properly is not to chase benefits, but to notice what happens when a plant evolves to protect itself with extreme chemical precision, and how that precision intersects with the long-term maintenance systems of the human body. Cloves come from the dried flower buds of a tropical evergreen tree, harvested just before the flower opens. This timing matters. At that stage, the plant concentrates defensive compounds designed to protect its reproductive potential. From a biological perspective, these compounds exist to deter insects, inhibit microbial growth, and resist oxidative damage in a warm, humid environment. When humans consume cloves, they are not borrowing a nutrient in the conventional sense, but interacting with a chemical defense system refined by evolution. The dominant compound in cloves is eugenol, a phenolic molecule that is responsible for their characteristic aroma and most of their biological activity. Eugenol is not a vitamin, not a mineral, and not a macronutrient. It functions primarily as a regulator of chemical reactions. In the context of longevity science, this distinction is crucial. Aging is not driven by deficiency alone. It is driven by accumulated dysregulation, oxidative stress, low-grade inflammation, and loss of signaling efficiency. Compounds like eugenol matter not because they add something new, but because they influence how existing systems behave over time. Oxidative stress is one of the most studied contributors to biological aging. It arises when reactive molecules damage lipids, proteins, and DNA faster than the body can repair them. Cloves consistently rank at the extreme high end of antioxidant capacity when measured in vitro. This fact is often repeated without context, so it is worth slowing down. High antioxidant capacity in a test tube does not mean direct lifespan extension in humans. What it does indicate is that the compounds in cloves are structurally suited to donate electrons and stabilize reactive species. Eugenol's molecular structure allows it to interrupt chain reactions that propagate oxidative damage. More interestingly, eugenol does not act only as a scavenger. It also influences endogenous antioxidant systems by modulating enzymes involved in redox balance. This dual role, direct and indirect, aligns with what modern longevity research increasingly values. Longevity is not about overwhelming the body with antioxidants, but about supporting its ability to regulate oxidative processes efficiently. Inflammation is another core process that links aging, metabolic dysfunction, and chronic disease. Unlike acute inflammation, which is protective, chronic low-level inflammation gradually damages tissues and disrupts signaling. Clove-derived compounds have been shown to interact with inflammatory pathways by influencing mediators such as prostaglandins and cytokines. Again, the key is not suppression, but modulation. Eugenol appears to reduce excessive inflammatory signaling without shutting down necessary immune responses. This subtlety matters. Systems that are overly suppressed become fragile. Systems that are regulated remain adaptive. From a longevity perspective, this distinction separates compounds that appear powerful in the short term from those that support resilience over decades. The relationship between cloves and metabolic health provides another layer of insight. Metabolic aging is characterized by reduced insulin sensitivity, impaired glucose handling, and altered lipid metabolism. Research involving clove extracts suggests that certain compounds can influence how cells respond to insulin signaling. The effect is modest, context-dependent, and far from a treatment, yet it is consistent. Clove compounds appear to enhance cellular responsiveness rather than forcing glucose reduction. This aligns with a broader principle in longevity science. Long-term metabolic health depends less on lowering numbers and more on maintaining responsiveness. A system that responds appropriately to signals ages more slowly than one that requires constant correction. Microbial balance in the gut and oral cavity is another domain where cloves show interesting behavior. Eugenol exhibits antimicrobial activity against certain bacteria and fungi, particularly those associated with spoilage and infection. Historically, this property made cloves valuable for food preservation and dental care. In modern terms, this activity raises an important question. Can antimicrobial plant compounds support microbial balance without causing disruption? The evidence suggests that clove compounds are selective rather than broadly destructive. They tend to inhibit pathogenic organisms more strongly than beneficial commensals. While this area is still under investigation, it highlights a recurring theme. Plants evolved chemical tools to manage microbial environments, not sterilize them. When used in small, dietary amounts, these tools may support balance rather than dominance. The nervous system is often overlooked in discussions of spices and longevity. Yet, it is one of the most vulnerable systems to cumulative stress. Eugenol interacts with ion channels involved in nerve signaling, which explains its traditional use in dentistry as a local analgesic. This interaction does not imply systemic pain relief, but it does suggest an ability to influence neuronal excitability. Over time, excessive excitatory signaling contributes to stress, poor sleep, and neuroinflammation. Compounds that gently modulate neural signaling without sedation may play a role in long-term neurological resilience. This is not about calming the mind in a psychological sense, but about preserving the integrity of neural communication under stress. What makes cloves particularly interesting is not any single mechanism, but the convergence of multiple small effects acting on different systems. Oxidative regulation, inflammatory modulation, metabolic signaling, microbial balance, and neural interaction all intersect with aging biology. None of these effects are dramatic. None promise transformation. That is precisely why they matter. Longevity is rarely influenced by interventions that act loudly. It is shaped by inputs that are quiet, consistent, and system-aware. Dose and context are essential considerations. Cloves are potent. Their concentration of bioactive compounds means that more is not better. In culinary traditions, cloves are used sparingly, often combined with fats and other spices. This pattern likely reflects an intuitive understanding of potency long before modern chemistry. Fat enhances the absorption of phenolic compounds, while combination reduces dominance. From a systems perspective, this mirrors how the body prefers inputs: balanced, contextual, and moderate. Extracts and supplements remove this context, which is why their effects are less predictable. Longevity science increasingly recognizes that whole food matrices behave differently from isolated compounds. Another overlooked aspect is the role of sensory perception. The strong aroma and taste of cloves activate olfactory and gustatory pathways that influence digestion, satiety, and even hormonal responses. Sensory input is not superficial. It primes the nervous system and digestive processes. Foods that engage the senses tend to produce more coordinated physiological responses than neutral inputs. Over a lifetime, This coordination may contribute to metabolic efficiency and digestive resilience. Historically, cloves were valued not only for flavor but for preservation and protection. These uses were not symbolic. They were functional responses to environmental challenges. In warm climates, microbial growth and oxidative spoilage are constant threats. Plants that could resist these threats survived. Humans who incorporated such plants into their diets benefited indirectly. Modern refrigeration and sanitation have distanced us from these pressures, but our biology has not changed. We still age through the accumulation of oxidative and inflammatory stress. The relevance of cloves is not nostalgic. It is biological. From a modern longevity perspective, cloves represent a category rather than a solution. They exemplify how small, potent plant compounds can influence regulatory systems without overwhelming them. They remind us that aging is not a single process to be stopped, but a network of processes to be supported. When viewed this way, cloves are not a hack. They are a signal that long-term resilience often comes from inputs that operate at the level of regulation rather than force. The temptation in longevity discussions is always to search for the strongest effect, the fastest change, the most impressive metric. Cloves resist this narrative. Their value lies in their restraint. They do not push the body into a new state. They support the body's ability to remain in balance. Over time, balance is not static. It is dynamic and adaptive. Compounds that respect this dynamic nature are more compatible with long-term health. In practical terms, incorporating cloves into a dietary pattern is less about frequency and more about consistency. Small amounts, used regularly, within meals, reflect both traditional usage and modern understanding. This approach minimizes risk while allowing subtle effects to accumulate. Longevity is cumulative by nature. It responds to patterns, not events. When scientists study populations with long lifespans, they rarely find a single ingredient responsible. They find patterns of eating that emphasize variety, moderation, and the regular presence of bioactive plant compounds. Cloves fit comfortably within this pattern. They do not dominate it. They contribute to it. Ultimately, the story of cloves and longevity is a story about scale. Small inputs acting over long time frames, mild effects integrated across systems, chemical precision refined by evolution meeting biological systems shaped by adaptation. Understanding cloves in this way shifts the conversation from what they can do for you today to how they quietly participate in the maintenance of the systems that carry you forward over time. If longevity is the art of staying functional for as long as possible, then cloves are not a shortcut. They are an example of how attention to subtle mechanisms, respect for biological context, and consistency over intensity align more closely with how the body actually ages. That perspective is not exciting. It is something better. It is accurate. There is another layer to cloves that rarely enters longevity discussions. Yet it matters if the goal is to understand aging as a systems problem rather than a checklist of nutrients. Aging is not only biochemical, it is also regulatory. Over time, the body loses precision, signals become noisier, feedback loops become slower, small errors compound. When researchers talk about biological age rather than chronological age, they are often describing how well these regulatory systems still coordinate with one another. Cloves are interesting here because their compounds tend to act upstream, at points where regulation is decided, rather than downstream where damage is already visible. Take cellular stress responses as an example. Cells constantly monitor their internal environment and adjust gene expression in response to stress. Certain plant polyphenols, including those found in cloves, appear to activate mild stress response pathways without causing harm. This phenomenon is sometimes described as hormesis. The idea is simple. A low-level challenge encourages the system to maintain its adaptive capacity. Over time, systems that are never challenged become fragile, while systems that are overwhelmed break down. Clove compounds seem to sit in that narrow middle ground, providing a signal without forcing a response. From a longevity perspective, this is more valuable than blunt intervention. Mitochondria deserve special attention in this context. These organelles are not just energy producers. They are central hubs for metabolic signaling, oxidative balance, and programmed cell responses. Mitochondrial dysfunction is a consistent feature of aging across tissues. Some evidence suggests that phenolic compounds like eugenol can influence mitochondrial efficiency by reducing oxidative burden and supporting membrane stability. This does not translate into more energy in a simplistic sense. It translates into less waste, less leakage, and more predictable output. Over decades, predictability matters more than peaks. Another overlooked dimension is vascular health. Aging blood vessels gradually lose elasticity, impairing nutrient delivery and waste removal. Oxidative stress and inflammation accelerate this process. Clove-derived compounds have demonstrated effects on endothelial function in experimental settings, primarily through their interaction with oxidative and inflammatory mediators. The significance here is not acute improvement, but preservation. Blood vessels that maintain responsiveness support every other system. Longevity is not compartmentalized. When circulation declines, everything declines with it. There is also an important behavioral dimension that rarely appears in scientific papers, but consistently appears in real populations. Ingredients like cloves shape eating patterns. Their strong flavor encourages slower eating, smaller portions, and more intentional food preparation. These behaviors influence hormonal responses related to satiety and digestion. They also reduce metabolic volatility. Over time, reduced volatility means less stress on insulin signaling, less inflammation, and more stable energy regulation. Longevity is influenced not only by what compounds do in isolation, but by how they shape habits. Cloves also highlight an uncomfortable truth in modern nutrition science. Many of the compounds most relevant to long-term health are not essential nutrients. You do not develop a deficiency disease without them. This makes them easy to dismiss. Yet aging itself is not a deficiency disease. It is a gradual loss of coordination. Compounds that preserve coordination rarely announce themselves dramatically. They work quietly, often below the threshold of clinical significance in the short term. Cloves fit this profile precisely. The safety profile of cloves reinforces this perspective. Used in traditional culinary amounts, they have been consumed for centuries across cultures without evidence of harm. Problems arise primarily when context is removed, such as with concentrated oils or extracts used excessively. This pattern is instructive. Longevity-friendly inputs tend to be those that integrate smoothly into daily life rather than override it. The body responds better to signals it can contextualize. It is also worth noting that cloves rarely act alone. In traditional cuisines, they are paired with other spices that share overlapping but distinct chemical profiles. This creates redundancy and balance. From a systems biology perspective, redundancy is protective. When multiple inputs support similar pathways through different mechanisms, the system becomes more resilient. Longevity is resilience extended over time. Modern research increasingly supports the idea that dietary patterns rich in diverse plant compounds correlate with slower biological aging. This correlation persists even when calorie intake, macronutrient ratios, and lifestyle factors are accounted for. The implication is that chemical diversity matters. Cloves contribute disproportionately to this diversity because of their potency. A small amount introduces a wide range of bioactive interactions. The temptation, especially in longevity-focused media, is to elevate one ingredient above all others. That approach consistently fails. Biology does not reward obsession. It rewards coherence. Cloves make sense when they are understood as part of a broader pattern of low-level consistent support for regulatory systems. If there is a single takeaway worth holding on to, it is this: longevity is not extended by fighting aging aggressively. It is extended by reducing the rate at which small errors accumulate. Cloves do not repair damage. They do not reverse time. They appear to reduce the background noise against which the body must constantly operate. Less noise means clearer signals. Clearer signals mean better decisions at the cellular level. Over a lifetime, better decisions matter. Understanding cloves in this way reframes their role entirely. They are not functional food in the modern marketing sense. They are not supplements disguised as spices. They are an example of how evolutionary chemistry and human physiology intersect quietly, effectively, and without drama. That is why cloves are relevant to longevity, not as a headline, but as a footnote that turns out to be more important than it first appears. To fully appreciate why cloves remain relevant in longevity discussions, it helps to zoom out and look at aging as a problem of information loss. In youth, biological systems exchange information efficiently. Cells respond accurately to signals. Tissues coordinate with one another. Repair mechanisms activate when needed and stand down when the job is done. Aging, at its core, is the gradual erosion of this informational clarity. Signals arrive late, responses overshoot or fall short, and feedback loops drift out of alignment. The result is not immediate failure, but inefficiency that compounds year after year. Plant compounds, like those found in cloves, interact with this informational landscape rather than overriding it. Eugenol and related molecules influence transcription factors, enzyme activity, and membrane dynamics. These are not endpoints. They are control points. When control points remain responsive, systems retain flexibility. Flexibility is one of the strongest predictors of biological youth. Rigid systems break. Flexible systems adapt. One area where this becomes especially relevant is immune aging. As the immune system ages, it tends to become simultaneously weaker and more inflammatory. This paradox is one of the defining features of late-life vulnerability. Low-grade inflammation persists, while effective responses to new challenges decline. Clove-derived compounds appear to influence immune signaling in a way that nudges the system toward balance. They do not stimulate immunity in a general sense. They appear to dampen excessive signaling while preserving responsiveness. This distinction matters deeply for longevity, because chronic immune activation accelerates tissue damage, while immune suppression increases risk from external threats. There is also growing interest in how dietary compounds affect epigenetic regulation. Epigenetics refers to chemical modifications that influence which genes are expressed without changing the underlying DNA. These modifications respond to environment, stress, and nutrition. Over time, unfavorable epigenetic patterns accumulate, contributing to metabolic dysfunction and age-related decline. Some phenolic compounds have been shown to interact with enzymes involved in epigenetic marking. While this research is still developing, the implication is important. Longevity may depend not only on protecting DNA from damage, but on preserving the flexibility of gene expression. Cloves, through their influence on regulatory enzymes, may contribute modestly to this flexibility. Another subtle but meaningful dimension is digestive efficiency. As people age, digestion often becomes less effective, not because of reduced intake, but because of reduced coordination. Enzyme secretion, bile release, and gut motility become less synchronized. Strongly aromatic spices like cloves stimulate digestive signaling through sensory pathways that begin before food even reaches the stomach. This anticipatory signaling prepares the digestive system, improving efficiency without increasing demand. Over decades, improved efficiency reduces metabolic strain and nutrient mismanagement. Sleep quality is another indirect pathway worth considering. Longevity research consistently links sleep integrity with slower biological aging. While cloves are not sedatives, their influence on neural signaling and inflammation may support sleep indirectly by reducing background stress signaling. Chronic low-level inflammation and oxidative stress interfere with sleep architecture. Inputs that reduce this interference, even slightly, can have outsized long-term effects. What emerges from this perspective is a consistent pattern. Cloves do not act loudly in any one domain. They act quietly across many. This distributed influence is precisely what makes them compatible with long-term health. Aging is rarely driven by a single failing system. It is driven by many systems drifting together in the wrong direction. Inputs that touch multiple systems gently may be more valuable than those that target one system aggressively. This also explains why cloves resist simplistic narratives. They are difficult to market as miracles because their effects are contextual and cumulative. Yet this is exactly how longevity works in real biological systems. Longevity is not an event, it is a trajectory. There is a tendency in modern health culture to separate food into categories of pleasure and function. Spices like cloves blur this distinction. Their function is inseparable from their sensory presence. This integration matters. Humans evolved in environments where survival and sensory engagement were not separate. Foods that engage the senses often carried information about safety, potency, and preservation. Ignoring this dimension impoverishes our understanding of nutrition. From a practical standpoint, the most longevity-aligned use of cloves is unremarkable. Incorporated into regular meals, combined with other whole foods, and used consistently but sparingly. This pattern avoids peaks and troughs in exposure. It mirrors how biological systems prefer to be supported, slowly, repeatedly, without shock. When researchers look back at populations with extended health span, they do not find extreme intakes of any single compound. They find cultural patterns that unintentionally optimize for regulation. Cloves appear repeatedly in such patterns, not as stars but as constants. Constants matter. In that sense, cloves offer a useful lens for evaluating longevity advice more broadly. If an intervention promises rapid change, dramatic results, or singular importance, it is likely misaligned with how aging actually unfolds. If an intervention supports regulation, balance, and resilience without demanding attention, it is worth considering. Cloves pass this test quietly. Their relevance to longevity is not about extending life in a measurable way within a short study window. It is about participating in the slow work of keeping systems responsive, coordinated, and resilient in the face of time. That work does not generate headlines. It generates stability, and stability, over long enough time frames, is what longevity really means.